Welcome to Lecture 2.4 on Molecular Biology and the Regulation of Gene Expression. I'm Wendy Nofke and I will be giving your presentation today. Gene expression is the term we use for using a gene to make a protein. Since we have many genes and many different proteins, we need to have a way of being able to regulate this. Regulatory proteins can actually bind to regulatory elements, which are regions of the DNA near the promoters. These can interact with the enzyme RNA polymerase and help determining when you're going to actually express the gene and when it will be turned off. Activators can enhance interactions of the RNA polymerase with the promoter, while repressors can impede the progress of the RNA polymerase along the strand. Prokaryotic gene regulation is a little bit simpler than in eukaryotes. Prokaryotes will form operons, which are regions of DNA with one or more genes that will encode proteins that work together for a specific function. These will include a promoter and an operator. The operator is the region of the operon where the regulatory proteins will bind. A common example that's been studied a lot is the lac operon in E. coli. This is going to regulate the production of the enzyme lactase. With this operon, lactase is only produced when lactose is present from the environment. When lactose is not present, it will actually be shut off, and this is a way for the cell to save energy. Eukaryotic regulation is more complicated. There's many different regulatory proteins and elements involved. They may involve enhancers, which are distant regions of the DNA that can actually loop back and interact with the promoter. One example of regulation in the eukaryote is the TATA Box. The regulatory element that is part of the promoter of most eukaryotic genes is the TATA Box. There's a lot of regulatory proteins that will actually bind the TATA Box. All regulatory proteins must be present for the RNA polymerase to actually bind to the promoter. By having the TATA Box, this can allow multiple regulatory elements to be involved with the control. Another example is that cells need to be regulated particularly closely during development. The genes need to be turned on at just the right time in order to develop organs and organ systems, and then be shut off so that you don't develop too many of them. The homeobox genes are going to code for regulatory proteins that help to control series of developmental genes. And in this illustration here, you can see genes that will be color-coded to code a series of structures for this insect. Gene expression is also related to cancer. Usually, mutations causing cancer occur in the regulatory genes. Initially, the mutation inactivates tumor suppressor genes. This allows the cells to proliferate. The mutation will inactivate repair in the cells. The proto-oncogene, which is the normal version of a gene, will mutate to become the oncogene. The mutation then inactivates more tumor suppressor genes, eventually leading to cancer. Most cancers do occur in older individuals because it usually takes time for enough of these mutations to occur to actually result in cancer.